Hi, my name is Buddhadev Layek, an assistant professor at North Dakota State University. This is a collaborative project between North Dakota State University and Sanford Health. So as you know that the lung cancer is the leading cancer killer in both men and women, which is account for more than 25% of all cancer death. Moreover, a large proportion of the patients are diagnosed with metastatic diseases, and therefore the 5-year relative survival rate is only 6%, resulting in a greater than 50% of the mortality rate within a year of diagnosis. However, the current chemotherapeutic approaches for lung cancer extend life by only a few months compared with the best supportive care in lung cancer. The poor availability of the chemotherapeutic agent in the deep-seated and metastatic cancers is the main reasons for the poor therapeutic efficacy, which often leads to the drug resistance. Therefore, the tumor-targeted delivery can enhance the therapeutic efficacy while minimizing the non-specific toxicity of the anti-cancer drugs. Furthermore, the combination therapies targeting different key signaling pathways might be needed to overcome the drug resistance and achieve durable clinical responses. Therefore, the purpose of our project is to advance the mesenchymal stem cells, which is abbreviated as MSC, as a drug delivery vehicle for the tumor-targeted delivery of paclitaxel and entinostat combination to treat the patient-derived xenograft model of non-small cell lung carcinoma. So this is the central hypothesis of our project. So what we did initially, we cultured the mesenchymal stem cells, which we purchased from the science cells, and then we load these mesenchymal stem cells with the drug-loaded nanoparticles by incubating them with the MSC, and after the incubation, the MSCs are actually trypsinized and collected by centrifugation, and they injected into the mouse. When they injected into the mouse, due to their tumor-tropic behavior, the mesenchymal stem cells go to the tumor sites and slowly release the nanoparticles or the free drugs from the MSC. So let's go through the different methodology and the results. So here, initially we check the synergistic potential between two drugs. One is the leading chemotherapeutic agents, which is approved for the lung cancer treatment, paclitaxel, and we selected a HDAC inhibitor, entinostat, as the second drugs. So what we did to determine the synergistic potential between these two drugs, so we cultured the A549 cells, which is a human lung adenocarcinoma cells, and we treat these cells with a different combination of paclitaxels and the entinostat. And after 3 days of treatment, the cell viability is determined by the MTTSA. And when we get the viability, we actually plug this data into the Combinifet software that actually help us to determine whether this combination have the synergistic or additive effect or they are antagonistic. So as you look into this plot, so here I can tell you that if you see something boxes which is more like blue in color, that means there is a potent synergy. But on the other hand, if you see something the deep red, that means that particular combination has the antagonistic properties. And this is actually the surface surface plot of the synergy and antagonism. So if you see something is elevated, that means they have the that particular combination is have the synergistic potential. So this data actually shows that the paclitaxel and entinostat have a synergy over a wide range of combination. That's actually good. So then we actually determine why these two drugs have the synergistic potential. For that, as I mentioned that the entinostat is an HDAC inhibitor. So that means it will increase the acetylated histones in the cells. So here what we did, we treat the A549 cells with entinostat and paclitaxel, and paclitaxel is not an HDAC inhibitor. So you can see that when the drugs are treated with entinostat, there is significant increase in the amount of acetylated histone for both H3 and H4. But when the drugs are treated with paclitaxel, cells are treated with paclitaxel alone, there is no increase in acetylated histone. But when the cells are treated with both paclitaxel and entinostat combination, surprisingly, the amount of acetylated histone is increased. That actually tells that in presence of paclitaxel, the HDAC inhibition activity of the entinostat is increased, and that could be the potential reason for the synergism between these two drugs. Now, so we determine that these two drugs have the synergy when they actually added together. So now our next aim was to encapsulate this drug into the nanoparticles. For that, we use the PLGA, which is an FDA-approved polymers, and we individually load these two drugs in the nanoparticles. So the purpose of loading these two drugs individually is that so that we can control their release rate separately, and we could like change the dose of the drugs accordingly. So as you see that both the drug-loaded nanoparticle have a size around 208 nanometer, and both the carry the negative surface charge, which is actually good. And then their drug loading capacity is also very high. For paclitaxel, which is about 18.1%, and for entinostat about 14.5%. And you can see their release rate. Both the drug loaded nanoparticle shows the sustained release profiles. So our next aim was to load this nanoparticle in the MSC, and after that we'll like this drug loaded or nanoparticle loaded MSC will inject into the tumor bearing mice to check their efficacy. So before like putting this like nanoparticle into the MSC, we actually modified the mesenchymal stem cells. We actually cultured these mesenchymal stem cells in presence of azide containing sugar, so that they actually like synthetically produce some like azide moiety on their surface, and then this azide moiety we can use to load the nanoparticles, because in that way we can load this MSC with more amount of nanoparticle. Some of the MSC, some of the nanoparticle can be taken up by the endocytosis, and the other of the nanoparticle can be decorated on the surface due to this like conjugation of the DBCO conjugated nanoparticle with the azide groups through the click chemistry. And we have found that when these like nanoparticles are incubated with azide level MSC, after 4 hours of incubation, for paclitaxel, we saw that about 55 microgram of paclitaxel present in 1 million of cells. Similarly, for entinostat, we found about 48 microgram of entinostat per million of the MSC. Now, one of the interesting thing about this project is that actually we have a good collaborator at Stanford. We found Dr. Cornelius Dyke, who is the PI of this project also, and he is a amazing scientist and a doctor, like surgeon. So he provide us the different like lung tumor patient tissues from the Stanford Health. And then this tissue we collected and then we cut into the small cubes of about 2 millimeter and then we implanted into the mouse. And we are able to generate like a different like PDX model of the lung tumor. Out of the 6% we actually able to grow from the 3% which are currently on different phases of propagation. And some of these PDX mouse, which are in generation 3, we are now using for our in vivo anti-tumor efficacy. And these are the different groups. We use like PDX nanoparticle, means paclitaxel loaded nanoparticles. Then we use antinostat loaded nanoparticles and the combination of the nanoparticles. And we use also the MSC loaded with paclitaxel nanoparticle, then MSC loaded with antinostat nanoparticle, and then the combination of MSC loaded with paclitaxel nanoparticle and MSC loaded with ENT nanoparticles. As you see that this is still in initial phase of the study. We just only able to collect the 3 weeks of data. This study is currently continuing at like NDSU in Department of Pharmaceutical Sciences, Animal House. So you can see that the current trend, the initial trend is the combination of MSC significantly inhibited the PDX tumor growth in compared to the other groups or other treatment groups and as well as the control. Therefore, we can conclude that the combination of the paclitaxel and antinostat could be a potential like regimen for the successful treatment of the lung cancer. And with that, I would conclude my presentation, and I would like to thank my collaborator, Dr. Cornelius Dyke from Stanford Health, for being such an amazing collaborator and helping me in this project and with his suggestion or like that. And I also like to thank the DaCCoTA CTR pilot grant for funding this interesting project. And with this, I would like to thank you, everyone, and have a great day. Bye.